the next presentation will be by Natalia Miscalata for the company, which is AAVantgarde Bio.
So my name is Natalia Michatelli. It's a hard one.
Sorry
about that. I actually have two more surnames, so I spared you the Montenegro Soranzo. So I'm being kind, really. So we've heard a lot. We've heard a lot about gene therapy, and this is another gene therapy. But it's tempting to sort of start switching off. But I ask you not to, because this is actually something new and a scientific breakthrough. So therefore, let me tell you more about what we're doing. So we're focused on IRDs. We are a gene therapy company, and we are treating the root cause of the disease. So we're doing gene augmentation. And that is exactly what Luxturna obviously did. But what we're doing is we're treating diseases where the gene is too large to fit into a standard AAV cassette. So I think one of my American speakers called it zooming out, so I'm trying a new word. So zooming out, there's about 4,000 genes that are too large for a standard AAV cassette. So this is not just an ophthalmology play, but this is something that could be applied in other diseases as well. What we can do is, using our gene therapy approach, we have some very clever technology that was developed by our founder, Alberto Auricchio, who figured out how to do splicing technology. So basically, we can recombine the gene into a full-length, either full-length gene or full-length protein. So that's the background. We're starting with USH1B, and we're following on with Stargardt's, and I'll take a little bit of time and talk about both of them, but I promise you that I'm not going to take more than my 5 minutes. So I've got one slide on USH1B, and I can see in the audience that there are some of our PIs here in the room, so thank you for being here. We've treated 12 out of our 15 patients in phase 1/2. And because it's the first time that a dual AAV vector has been injected into the retina, we, much as one of our earlier speakers, have made sure that we have shared that data as early as possible to really share with the field that this is something that is feasible. We've got very nice safety data already that we've shared, and you can see there that multiple ophthalmology conferences, we've shared the data as it's been evolving. We will have completed the full enrollment by the end of this year, and we've dosed 12 out of 15 patients to date. Those 12 patients are in 3 different dose cohorts in terms of vector load, and so far we're seeing no issues in any of the dose levels. In addition to that, those of you who are following the field will have probably seen some of the presentations throughout the year where we're showing that we've got a nice first signal, but there's more to come. We believe that the data set needs to mature, so I'm not going to start showing you more data at this point in time, but we will be publishing, and there is more to come. Of course, we've also been engaging with the community, and we feel, similar to the prior presenter, that it's really important to get the feedback from the patients, and especially for the USH1B patients, that not only are struggling with vision loss, but also have impacts in terms of vestibular function and hearing issues to contend with. So it makes the trial actually much more complicated to do, but of course it also makes it's a disease area that's very needed a treatment. Our second program builds on some of the bones of our first program, which is really using the dual AAV technology. Actually, we're using AAV8, which has been mentioned in one of the prior presentations, so it's tried and tested. We know it's a safe capsid. And what we're using here is a similar technology to our first program for USH1B, but for Stargardt's disease, for ABCA4. What I wanted to show you here was the preclinical data. I know that many mice have been cured, but the reality is that large animal data does matter. So I wanted to just pause and show you the data before we go into the clinic. And we've got IND approval and CTA approval, so there will be more to come on this. But I just wanted to share the preclinical data set because it's so important for you to be aware of. We're treating the root cause of the disease, and what you're looking at is a very, very robust set of safety data. We've got mice 14 months data, which means the entire duration of a mouse that we've looked to see, is there any issues with our technology? That's really looking at, could there potentially be any issues with the splicing? And the same for NHPs. We've run 2 long-term NHP studies. But the most interesting, of course, is the efficacy data, and that's what you're looking at here. And we're the only ones in Stargardt's disease that's shown with knockout pigs that we can normalize. So when you're looking at the chart, you're at the left, you can see the control animals at 100% expression level, the control animals with 0, and then the treated animals are greater than the normal knockout pigs. So what you're really looking at here is the potential to normalize the expression levels, and that's really important. We've done the same in NHPs as well, in case somebody was wondering about whether or not there was a great difference between the knockout pig data and the NHPs, and we're seeing similar data there as well. So as we go into the clinic, we're really excited about the potential to really normalize for these patients and treating the root cause of the disease. And I'll just finish on this last slide, which is really the pipeline for AAVantgarde. So USH1B, our first dual AAV technology in the clinic, almost complete enrollment. Stargardt's disease, we're just starting to get going there. And behind that are other RRDs. That's it.